Good morning, children. Hope you have understood yesterday's topic. So, where we discussed a lot of things about cell and also the discovery of the cell and how it was named. And also, we have seen the matchstick experiment. The matchstick experiment, that is also we have seen. So, now today we are going to see how we can see the cells on our own without depending on anybody. So we will try to know that the organisms are made up of cells, or if we take any part of any body, that particular thing is made up of cells. We'll try to know these things on our own. For that reason, what we will do is, first of all, that is what we have done. That is what we have done yesterday also. We have taken a matchstick. We removed a thin slice of that and we observed it under the microscope, and we were able to see some cells in that. Now today also we are going to see another experiment where we are going to take the onion peel. What we are going to take is the onion peel, and with the help of onion peel we will see the cells on our own. Now what we have to do is, we have to take an onion and then we have cut the onion into some pieces, and we have to take one particular piece of that and then we have to under it under the microscope. But if you observe it directly, you will not be able to see anything, because the onion peel which you are taking is a bit thick and also it is opaque. So even in the microscope, when the light falls on the slide which you keep here, or onion which you keep here, if it is opaque, the light cannot pass through it. Only if the light passes through that, we will be able to see the reflection of that, and then you will be able to see the cells in that. Otherwise you cannot see that. For that reason, what we'll do is, we will remove the thin layer. If you have ever cut the onions, or if you have seen the onions, and if you try to open that, so when you cut it, you can see a thin peel in the onion. For example, if you see here, I am cutting it into two pieces, and what I am seeing here is, I am seeing here a thin peel. This is the onion and this is the peel which I have removed. Can you see the onion peel here? Okay, so this is the onion peel. Now you can see this is a transparent layer. It is an absolutely transparent layer. If we use this layer and check it under the microscope, you will be able to see the cells. But what is the process? How we have to check the onion peel? For that reason, first of all, we'll take a slide. If you go to a bookshop and all, you can find the slides like this, and in the slide, these slides will be glass slides, so that they can reflect the light. And then there are some round, circle or square shaped thin glass structures which are called as the cover slips. So what are needed is a slide, a glass slide, and a cover slit, and also glycerin, and also a little amount of water. So these are the things which are required so that you can make a slide. So first of all, what we'll do is, you'll take a slide like this. We'll make sure that don't touch the slide in the middle like this. So directly catch the slide in this way. Catch the slide like this. Don't catch the slide like this. Don't handle it like this. Only handle it like this, so that it will not receive any fingerprints, so that it will become a hurdle, it will become an obstacle, disturbance for viewing the cells. So that always hold the slide like this, okay? And then what we'll do is, we'll keep the onion peel on the glass slide. Then take this cover slate. Okay, before that, pour one drop of glycerin, or you can pour one drop of water, and make sure that the water or the glycerin will not have any kind of air bubbles, because if there are air bubbles, that again becomes an interruption to watch the cells. So because those will look very bigger in size, and that they will occupy a lot of space on the area where we are going to check the cells. So make sure that there will be no air bubbles on that. And once you keep the covers, once you keep the onion, you place the cover slip on this. After placing the cover slips, if it is a rectangle one or a circle one, it's not a big deal. And then slowly, without disturbing, take it and observe under the microscope. So you have different lenses in the microscope, okay, and you can see the different lenses here. See the place where you are going to look into that, and you can see the glass which is emitting the light, and you can see the place where you are putting the slide, okay. And there are different magnification of lenses. According to that, you can check it out. So once if you check it out, you can see the cells which are in the onion peel. So once you do this one, you can check the cells which are in the onion peel. So you can see the picture of the cells which you are going to see in the onion peel like this. And if you do this experiment in your lab, in our school lab, definitely you can see this kind of structures very easily with the available microscope at your school level. So now this is what we can do. And the second one is now coming to how to observe the animal tissues. How to observe the animal tissue. If you take your cheek, your cheek will have very transparent layer, very thin layer on the cheek. So what we have to do is we have to take a spoon. We have to slowly scrape the inner part of the cheek, and you have to remove the very tiny layer out of that. And once you get that out, a small layer, this layer is nothing but the epithelial layer, the outer layer. So it will be transparent. So this layer you have to remove it, and again you have to do the same experiment. You go ahead with the same experiment, and without any second thought, you can see, without any doubt, you can see the cells which are in the cheek cells. There is a difference between the cheek cells and the onion cells. The placement, the size, okay, they are a bit different, but the materials which are in them will be absolutely same. So epithelial layer, if you take that and if you place on the slide, put some glycerin on that and put the cover slip and observe in the microscope, you can see these cells which are there in the cheek cells. Okay children, after learning about the two experiments, that is human cell and the cheek cell and also the onion peel, we will learn about Robert Brown, who is a great scientist in the year 1773 to 1858, right? So now after so many scientists have given the best contribution to the cell, to know about the cell, to know the integral parts about of the cell and all, and one of them is Robert Brown. So it is believed that Felix Fontana, okay, who lived from 1730 to 1805, he is a scientist who actually observed the nucleus of the cell for the first time. So there is a part which is called as nucleus in the cell. So that nucleus was first observed by whom? That is Felix Fontana, and he could not give a proper explanation of that. He observed this particular nucleus in the epithelial cells, of the human body, in the transparent layers of the human body. So he is the first one to observe that. But the complete credit goes to Mr. Robert Brown, because he observed several things, not one or two. He observed several things, and especially when he was observing the orchid leaves and all, he found some circular spots in that. So he observed some circular spots in that, which are a bit different from all the other organelles. So Robert Brown, when he was observing the orchid leaves, he found some circular dark spots in that. And he thought that this should be one of the very important part of the cell. And when he observed the same structures in the other things also, like he might have seen other some structures like leaves or some root parts or blood or something. So he might have observed the same kind of structures in the cells. So these particular circular spot was very much prominent in all the cells. So he thought that this should be the integral, or you can say significant or most important part of the cell, and then he named it as nucleus. This all happened in the year 1831. So this 1831 year is the year, it's after almost 175 years from the time when the nucleus was first observed. So nucleus was first observed in the year 1750 or something. So the nucleus was first observed, it was almost 175 years gap. In the year 1650, the nucleus was first observed, and after 175 years, Robert Brown was able to tell the whole world that there is a part called as nucleus, and then that is the prominent part of the nucleus. It is an integral part of the nucleus. So Robert Brown was the first person to see the nucleus and also name it. And when he was looking this in the leaf and all, he was able to trace out stomata in there. You can see in the picture in the text tube which is over here. So you can also see the stomata here and there. So stomata are the parts which are present on the leaf, which will help in the diffusion of gases. The carbon dioxide goes inside, oxygen comes outside. How? It doesn't have a nose like us, isn't it? So it has only stomata on the leaves, and those stomata will help to diffuse the gases. That is how they will take the oxygen inside, carbon dioxide outside, or the carbon dioxide inside, oxygen outside. Or carbon dioxide and oxygen for the process of photosynthesis, oxygen and carbon dioxide for the process of respiration. So remember children, Robert Brown is the one who discovered nucleus. Robert Hooke is the person who discovered cell. Okay, is it clear? Now Robert Hooke discovered the cell in the dead cork. Robert Brown discovered nucleus in the orchid leaves. So this is how the a great contribution was given by Robert Brown to the cell biology. Is it clear children? Okay, so tomorrow we'll see how to observe the nucleus in the cheek cells and also in the leaves, and then we'll go to the next topic. Okay, see you then. Until then, bye.